Welcome to the third part and the last part of the lecture series on CRISPR-Cas9 technology. And today we are going to discuss the applications of this genome editing tool. As we have discussed the basics of CRISPR-Cas9 technology, it is now become clear that genome editing as well as the gene therapy for treating diseases like cancer and many infections and genetic disorders can be performed or can be cured by using this technology. As we have discussed in the first lecture that the gene editing technologies which are designed based on the restriction enzymes, example the transcription activator like effector nucleases or the talins and gene finger nucleases or the ZFNs depend on binding of the protein to the DNA. So the disadvantages of those technologies, one of the disadvantages are the proteins which we need to design for each of the experiments, right? But in the CRISPR-Cas system, the Cas9 endonuclease is used for genome engineering, where the guide RNA that is guiding the Cas9 for cleaving the target sites based on the DNA to RNA hybridization. So we just need to design the guide RNA component, which is comparatively easier to do rather than the enzyme engineering. So now we will discuss about its applications and future perspectives. But before, let us first see like little bit about this endonuclease, the Cas enzyme, Cas9. So it is the major Cas nuclease. It is a DNA endonuclease and it possesses two RNA molecules, as we have already seen. That is the CRISPR RNA, CrRNA, and the transactivating CrRNA or the tracer RNA. This system, as also discussed, can detect and degrade any foreign nucleic acids. Cas9 has two domains, that is nuclease domain and the recognition domain. The nuclease domain comprises of two subdomains, two subnuclease domains, that is the HNH and the RUBC. As you can see here, so this is the nucleus and this is the target DNA. So the HNH domain in the nucleus, it cleaves the DNA sequence, which is the target sequence. That is complementary to the guide RNA. So this is the sequence which is complementary to the guide RNA. And the other domain of the enzyme, the RUBC, it cleaves the sequence that share non-complementary to the sequence in the guide RNA. That means the non-target sequence. So now you have cleavage or DNA breaks at both the strands. So that is the outcome. That means it's a creation of a double strand break with some blunt ends. Now genome editing by CRISPR-Cas9 system primarily involves the DNA double strand breaks generated at the target site. As already discussed, the repairing of this DSB is initiated by either of the two pathways such as homology directed repair or the HDR or non-homologous end joining. The here, okay. The NHEG, the non-homologous end joining, it induces insertions. It can induce insertion here, or it can also delete some part of it. So insertion and deletion mutations are possible, as you can see here, like as a result of NHEG. All right, so some portion here is missing and some added nucleotides are present here. So these NHAG, as we have already discussed in the first lecture, it can disrupt the total open reading frame. Okay, so HDR pathway, on the other hand, here the homology directed repair, it can generate specific nucleotide changes like single nucleotide change, or some large insertions are also possible, like the addition of a fluorophore or the tag. So the DNA repair template here, this is the donor template. It is actually containing a desired sequence which must be delivered into the cell like kind of a DNA of interest with the gRNAs and the Cas9. So the template, this template must contain the required gene edit, means desired sequences to be added or modified, but also it contains some additional homologous sequences. So these are the homologous sequences immediately upstream or downstream of the target. So if this is my target, so I need to have some homologous sequences, homology with this upstream or downstream of the target site. Okay, so these are known as the left and the right homology arm. Why this is needed? Because these homology arms will help in the homologous recombinations, like what is happening here. This gap will be filled up by the addition of the nucleotides which are complementary to this particular site. So this way we can say, for example, this is my target site additions, we can add this sequence complementary to this sequence here in the new DNA. So this is what is happening, the insertion or modifications. So you can also add some modified or altered nucleotides here. So this way, by homology directed repair and using the CRISPR-Cas9, CRISPR-Cas internuclease, you can add a new nucleotide or modify the existing nucleotide. To help the researchers in designing experiments, several databases have been developed to provide information on the Cas genes and CRISPR loci. For example, the CRISPR-SO2 can analyze the experimental results performed using the CRISPR-Cas tool. CRISPR-P can be used to identify and design the proper target site sequence. CRISPR-Seq or eCRISP can be used to design the guide RNA. Talking about the appropriate delivery tool for transferring the CRISPR Cas into the cells, the delivery of the Cas9 and gRNA can be carried out by nucleofiction, lipofectamine mediated transfection, polyethylenamine mediated transfection, as well as electroporation. But at present, the most reliable approach for CRISPR-Cas component delivery to the cultured cells is by the use of the viral vectors like the lentivirus, adenovirus, or the adeno-associated virus. Genome editing experiments based on this CRISPR-Cas system, it has been found that this technology has kind of enormous potentiality for gene therapy, like to modify or like to remove the default the mutated or the disease genes. One of the best example is the treatment of the cystic fibrosis, which was treated uh, by the help of this CRISPR-Cas and following the homologous recombination of the gene, the mutated gene is here is the cystic fibrosis transmembrane conductance regulator or the CFTR gene. So you can follow this homologous recombination pathway to correct these gene mutations, and that has been tried already. Huntington disease is another disease, the nervous system neurodegenerative disease, or the Duchenne muscular dystrophy. So they are actually can have been already tried, and the gene, the mutated gene can be corrected with the help of the CRISPR-Cas9 tool. It is also applied for editing the plant genome, such as, and has been tried on the wheat, rice, and several other plants, improving the crop production. Um, one more thing is that this CRISPR-Cas system is the inbuilt immunity for the microbes, for the bacteria, right? So we can exploit it, we can use it to make the bacterial cultures more resistant to the viral attack. So the genomes of this industrially important bacteria also can be edited for the high production of biofuels, anti-cancer compounds, and the antibiotics. It has got a great potential in the field of drug discovery and for the generation of therapeutic drugs for treating the genetic diseases. Uh, Genomic screens can be performed by the CRISPR-Cas9 system to detect the mutated genes that have become drug resistant, analyzing the activity of drugs on infectious agents, cancer cells, as well as proteins or genes involved. It can also help in determining the activity of a gene in any disease conditions, correcting the gene that causes harmful mutation and switching off or switching on some cancer causing genes or kind of switching on the tumor suppressor genes. So it can do a lot of things, right? Now, uh, let us, so we come to the last part of this lecture, and let us see how the scientists are using the CRISPR technology to design a rapid testing kit for COVID-19 diagnosis, which is very relevant nowadays. The system uses Cas12 instead of Cas9. It is not the kit that they have developed, it is based on Cas12. Why? Because Cas9 is very stringent and act like just a molecular scissor which after binding to the target site will cut the target DNA, only just make a cut. Whereas Cas12, another CRISPR associated endonuclease, is like a paper shredder, like it will just keep on degrading the DNA strand once it get activated by binding to the target DNA. So the Cas12 will start cutting without stopping like a paper shredder. The kit uses another SS, the single strand DNA probe. It is a reporter molecule tagged with a fluorescent dye. It is a fluorescein amidite, FAM, that produces a color, but the fluorescence of FAM is generally not observed in the intact probe due to the presence of a quencher molecule. Okay, so here uh, when the Cas12 is activated means when this binding is proper and the Cas12 is now active, and it will start to degrade the target DNA and whatever the single strand DNA present in the reaction mix. What will happen? It will also attack on this single strand DNA probe. Okay, so it will cut this DNA indiscriminately, as a result of which the probe will be also like affected. And say for example here, as we have seen, that the quencher will be removed, this part of the probe will be removed, and this fluorescence part is now free. It is now liberated and it can be visually detected by flow strips. So these are some flow strips, as you can see here as well as here. Since the probes are these probes are tagged with, so this is the streptavidin beads are also here. These due to the presence of the streptavidin, the flow strip first captured here. If the molecule is intact, the streptavidin associated, the full probe is intact, then it will be first captured here at this side, producing a thick band here. Okay, so here we have a biotin. Biotin will capture the streptavidin. Now, if the COVID-19 RNA is present and the Cas12, the CRISPR Cas12 that is active, and it is the Cas12, the active Cas12 is cutting this particular probe, then the streptavidin will not be attached here in this probe. So there will be no color formation at this region, because the color formation part is not attached to the streptavidin part. So the flow will go through, but here we have some antibody which will capture this part of the probe. Okay, so the color formation will be more concentrated over this part. So whoever is positive for the COVID-19 infection virus, the CRISPR, the Cas12, that system will capture the COVID-19 RNA. The Cas12 get activated, this will be cleaved, and then you will form a band over here. So this is how the CRISPR-Cas12 system can help in detection of the COVID-19 virus. So this is the representative strips. You can see the flow strips. You can see. So these are these bands. Is sorry. This is actually a little bit incorrect. So you have a band over here. So this will be positive. This will be negative. This is positive, negative, positive, negative. So the thick bands are present on these strips. Okay. All right. So this is how the CRISPR-Cas9 is nowadays is like rapidly. I mean, it can detect the COVID-19 presence of COVID-19 in a patient very rapidly. So this brings us to the end of this lecture series on CRISPR-Cas9 technology and its applications in different fields of research. More importantly, the detection of diseases like COVID-19. I hope you have enjoyed the lecture series and it was useful. On behalf of School of Life Sciences, I thank you for your participation and interest in these lecture series. Thank you.